Okay, C4 cycle, it is also called as a Hutch-Slack cycle because this has been first time explained by Hutch and Slack. C4 cycle is alternative pathway of Kelvin cycle, that is C3 cycle, taking place during the dark phase of the photosynthesis. In the C4 cycle, the first stable compound containing 4 carbons compounds, so that is why this has been named as C4 cycle. This particular C4 cycle having first carbon 4 carbon compound named as a oxalic acetic acid, so that is why OAA. Hence, it is C4 cycle name has given. It is a process of C2 fixation because it is also considered as a alternative cycle of that C3 cycle. The C4 pathway was worked out by Hutch and Slack in 1966. Hence, the C4 cycle is also called Hutch-Slack cycle. It is also termed as a C4 dicarboxylic acid pathway. C4 dicarboxylic acid pathway. It occurs in particular some plants like grasses, sugarcane, maize, sorghum, Atiplex, rosa, and amaranthus species, commonly containing this particular type of cycle. The plants exhibiting C4 pathway are called C4 plants. The C4 cycle exhibiting plants considered as a C4 plants. Till 1965, it was universally accepted that Kelvin cycle, that is C3 cycle, was the only pathway of CO2 fixation in the photosynthesis. But in the year of 1965, Hurst, Shack, Hart, and Burr. See here in 65, first term, this fundamentals of the C4 cycle has been described by 2 important scientists. Those persons' name are Korchak and Hart and Burr. These people first time given a fundamental information about this C4 cycle, and these people have been described and discovered C4 carbon pathway in sugarcane leaves, particularly when they were going to perform an experiment in the sugarcane plants. So that they were selected sugarcane leaves. In that leaves itself, these activities have been found. The C4 plants shows a specific types of leaf anatomy. We'll see one by one in this particular aspect of the studies. So C4 plants, the plants whose first product of photosynthesis containing 4 carbon compounds, that is why these are this is called C4 plants. Another one important point is there, which plants are exhibiting or following this C4 pathway in their carbon fixation. Such a plants considered as a C4 plants. So why these plants using this particular alternative mechanism for the fixing of the carbon means these plants usually growing in drought condition and high temperature condition, and here also nitrogen also in high content and carbon dioxide limitations will be there in that particular environment. They are going to exhibiting, they are additionally following this particular pathway alternate to the C3 cycle. Here we should have to be concentrate that C4 cycle is not particularly going to fix the carbon. Ultimately, the end product of the C4 pathway, again they should exhibit C3 cycle, then only the carbon fixation will take place. Means here the C3 cycle is ultimate. After completion of C3 cycle itself, carbon is going to be fixed. Otherwise, it is not possible to fix the carbon. Due to C4 plant are going to fix more CO2 as compared to the C3 plants having their KRANZ anatomy. So here, a particular anatomical structure we can see in these particular C4 plants itself, such a particular anatomy named KRANZ anatomy. So here C4 plants lost 277 molecules of water to fix per molecule of carbon dioxide as compared to C3 plants uses 833 molecules of water approximately at 30 degrees centigrade. They also avoid photorespiration process. Here, the photorespiration, another one part where it is going to be exhibiting the plants, particularly in case of that C3 cycle alternately. So here they also avoid the photorespiration process in this particular aspect. Let us see this C4 plants. Why plants use C4 mechanism? Means C4 having a enzyme fixed carbon compared to the C3 plants. Here a particular activity and their anatomical features are very different. And these can also fix oxygen during carbon fixation process. At the same time, both the activities also will take place here. And this particular fixation of the oxygen starts the process of photorespiration instead of carboxylation of substrate oxidized, and as a result loss and start the process of photorespiration also will take place. This process occurs due to dual activity of riboscope present in C3 plant, such as oxygenase and carboxylase activity. So let us see about this particular KRANZ anatomy. Their vascular bundles are surrounded by two rings of cells. So here we can see this particular diagram where it is also having a round bundle sheath cells also we can found, and as well as the mesophyll cells. These two can make this KRANZ anatomical structure. So this particular type of structure is considered as a the chloroplasts are dimorphic in nature. Very important. It is very important feature in this structure. Dimorphic natured chloroplasts are rarely found in the anatomy. So here, dimorphic means two types of chloroplasts are found in this KRANZ anatomy. In the leaves of these plants, the vascular bundles are surrounded by the bundle sheath cells. Here, bundle sheath cells are going to be surrounded to the vascular bundles. These bundle sheath cells are very large parenchymatous cells. These also containing a large parenchymatous cells in their structure. And these bundle sheath cells have chloroplasts. In this particular bundle sheath cells having a number of chloroplasts formed. The chloroplasts of bundle sheath cells are very large when compared with the other cells surrounded by many other cells. Also, we can see some of chloroplasts. But here, the chloroplasts are very large-sized chloroplasts found in these bundle sheath cells. And here, very important point, the chloroplasts which were going to found in these large bundle sheath cells. So there lack the grana. Grana is absent in this chloroplast and containing the starch in this structure. Grana is absent in these bundle sheath cells of the chloroplast. And the chloroplast in mesophyll cells. So here another mesophyll cells are present, in particular such of mesophyll cells. So here you can see these bundle sheath cells are here. These are the very large sized bundle sheath cells are present. These large sized bundle sheath cells and mesophyll cells are there. So in the bundle sheath cells, what happened? Chloroplasts are lacking of grana, and in the mesophyll cells, very smaller sized chloroplasts also found. Those are containing the grana. So here, bundle sheath cells lacking of the grana, and mesophyll cells containing the grana. So that is why the chloroplasts are differentiating their structure. So that is why dimorphic chloroplasts are present in this type of KRANZ anatomy. This peculiar anatomy of the leaves of the C4 plants is called KRANZ anatomy. These bundle sheath cells are bigger and look like a ring and wreath. That is why the KRANZ German name has been shown. KRANZ means in the German wreath. Hence it is called KRANZ anatomy. The C4 cycle involved in two carboxylation reactions, one taking place in the chloroplasts of the mesophyll cells and another is going to take place in the bundle sheath cells. There are 4 steps in Hasselbalch cycle. They were found like particularly for first is carboxylation, second is a breakdown, and third is a splitting, fourth is a phosphorylation. Let us see one by one here. So just now we have seen about this particular advantages of KRANZ anatomy, and so it is a main function of KRANZ anatomy to provide a site in which CO2 concentrated around Rubisco and avoiding photo respiration mechanism, which is also one of the important phenomenon is going to take place due to presence of this KRANZ anatomical structure. So here, whatever we have seen about these four important steps are going to take place in the C4 cycle. That is the first one by one step we can discuss here. First step, conversion of PEP into oxaloacetic acid. The first step is called conversion of PEP into oxaloacetic acid. In this step, CO2 is going to react with the phosphoenol pyruvic acid PEP to form oxalic acid. This reaction takes place in the presence of enzyme phosphoenol pyruvate carboxylase. Whereas in steps 2, conversion of oxaloacetic acid into malate. So in this step, second step, conversion of OAA means oxaloacetic acid into malate. Oxaloacetic acid is converted into malate first step, and this reaction is catalyzed by malate dehydrogenase. Whereas in third step, conversion of malate into pyruvic acid. Malate is transported into bundle sheath cell. In bundle sheath cell, it is converted into pyruvic acid by releasing CO2. Whereas fourth step, conversion of pyruvic acid into PEP. So pyruvic acid reacts with ATP and generated phosphoenol pyruvate. This reaction takes place in the presence of pyruvate orthophosphate dikinase. So let us see overall the C4 cycle. If you see here like this, two important cells here. It is a bundle sheath cell. This lower cell is considered as a bundle sheath cell. Upper cell is considered as a mesophyll cell. So let us start from here. You can see if once in atmospheric activity also takes place from here. Oxaloacetic acid here. This oxaloacetic acid will gives rise to malate here. Oxaloacetic acid. So here phosphoenol pyruvic acid here, PEP is there. From the PEP, this oxaloacetic acid is going to be formed in the presence of PEP carboxylase. The PEP carboxylase is going to be directly absorb this carbon dioxide from the environment conditions. So here, this carbon dioxide also carbonic anhydrases also involved in the absorbing of this particular aspect. And here, this oxaloacetic acid is going to give rise to the malate. So the malate dehydrogenase enzyme is going to be used here. And here NADPH is utilized for the completion of this reaction. This malate is going to transfer from mesophyll cell to bundle sheath cell. Here you can see the way is there. This malate is going to enters into the bundle sheath cell. In the bundle sheath cell, this malate is reacted with one enzyme, malic enzyme. So the malic enzyme, another one NADPH is going to produce, and carbon dioxide is whatever carbon dioxide is there, that is going to be captured, and that is going to be pushed into the Calvin cycle. So C3 cycle here, alternate cycle, you can see all of you. From here again the C3 cycle is going to be takes place. See, this is alternative cycle for the C3 cycle. Means here some due to the tropical condition and due to like whatever things we have been discussed with there, such conditions are going to be avoided by these plants. So that is why these are going to form like these types of one mechanism. This mechanism is alternate mechanism, and this can help this carbon dioxide pushing into the Kelvin cycle. Again, ultimately, definitely, must and should that Kelvin cycle is going to be complete. After completion of this Kelvin cycle, this carbon dioxide is going to be fixes, and this malate is going to be gives rise to the pyruvate. So here the pyruvate will form in the bundle sheath cells. This pyruvate is again enters into the mesophyll cell. You can see here it is enters into the mesophyll cell, and this is going to be gives a pyruvate phosphate dikinase. This can be reacted under the enzyme name is considered as a pyruvate phosphate dikinase. This under the pyruvate phosphate dikinase, this pyruvate is going to be used rise to PEP. Again, the PEP is going to be formed. Here, the ATP is utilized. So, and from ATP, one molecule of the ATP is utilized, and AMP will forms, and again another one ATP and AMP will going to reduce raise to ADP, and that can also helps in the observing carbon dioxide from the atmosphere. So all of you can just remember this mesophyll cell activities, whatever going on, and at the same time bundle sheath cells, whatever going to be takes place in this particular aspect. So here mechanism also you can see here mesophyll cells. This is the mesophyll cell and this is the bundle sheath cell. So here in PEP particular this phosphoenol pyrite carboxylases will take place from the atmosphere. Carbon dioxide is going to be absorbed, and this is going to be forms oxaloacetate with the 4 carbon of the oxaloacetate, and that is going to be converted into malate. There are also 4 carbon malate. It can be enters into the bundle sheath cell. So here you can see the same activity. The malate is going to give rise to 3 carbon. You can see here in malate having 4 carbons, and this malate is going to the same pathway with the 3 carbon. Name is pyruvate. 1 carbon from the malate, 4 carbon of the malate is 1 carbon is going to be donated to the Kelvin cycle. So in the form of the carbon dioxide that is going to be involved in Kelvin cycle. So that Kelvin cycle is after completion of successful Kelvin cycle, the sugars are going to be fixes, means sugars are formed in the vascular tissues. These can be afterwards transferred into the vascular tissues. Here, this pyruvate, 3 carbons of the pyruvate again enters into the this mesophyll cell. Again, that can going to be utilized as this one molecule of the ATP and reacts with ATP. And 3 same thing. The pyruvate having 3 carbon, so PEP also having 3 carbon. Again, from the atmospheric carbon dioxide, it will be absorbed. See. Again, that will form 4 carbons of the oxidized gas. See, PEP 3 carbons, and in the presence of PEP carboxylase enzyme, that can taking 1 carbon dioxide molecule from the atmosphere, so that becomes a 4 carbon of the oxaloacetic acid. And after this 4 carbons of the oxaloacetic acid is going to form 4 carbons of the malate, and malate is going to give rise to 1 carbon donated in the form of the carbon dioxide into the Kelvin cycle, and here the pyruvate is having 3 carbons. So here exactly clearly you can concentrate on the carbon number, how much it is containing the product, and what type of transformation and is going to be takes place in the mesophyll cell and bundle sheath cell. These 2 important aspects are going to be concentrated in this particular aspect. So let us just now have been discussed about C4 plants, common C4 plants, maize plants, sugarcane plants, millet plants, sorghum plants, and grass species. And besides of these also, we can see Atriplex rose plants and as well as Amaranthus species. Most of the Amaranthus species also going to be takes place this type of activity. So here let us see once again the C4 plants. Whatever important aspect of the C4 plants means C4 plants having a also considered as a Hasselbalch cycle in the mesophyll cells and Kelvin cycle in the cells of the bundle sheath cells will take place. Here you can see the most of this mesophyll cells, this C4 cycle is going to be takes place. C4 cycle, of course, in bundle, that is KRANZ anatomy will be there, but when compared with this mesophyll cell and the Kelvin cycle, mesophyll cell and as well as this these 2 activities when these are going to be takes place means like mesophyll cells and Kelvin cycle in the cell of the bundle sheath cell will takes place particularly. So there are 2 carbon dioxide acceptors are found in the C4 plants. That is a phosphoenol pyruvate in the mesophyll cells and ribulose 1,5-diphosphate in the cells of the bundle sheath cells. And carboxylation is catalyzed by both phosphoenol pyruvate carboxylase in the mesophyll cell and the ribulose diphosphate carboxylase in the bundle sheath cells. These 2 activities also takes place. The first stable product of the photosynthesis containing 4 carbon compounds, that is oxaloacetate. So the particular structure, anatomical structure is special structure found in the C4 plants. So that it is one of the important C4 pathway, which is considered as a alternative pathway of that